half a minute early today. Welcome to the lecture about the prediction of the effects of sequence variation. This lecture is one that is designed to be much bigger than the time we have. And I will just fast forward through some of the aspects and focus on some others. In the context from now on, we're going to have a couple of wrap-up presentations where I hope that you come with questions that we then can put into the exercise, into the exam. Sorry. So we want to predict the effect of variance. We want to predict the effect on life ultimately, and that in some sense, let's begin with a detour here. Who invented it? Evolution.
Well, Darwin found out that there is evolution, but didn't invent it.
If so, if the first part of it is true, if Darwin sort of invented it, or it started with Darwin, what's the Darwin-Wallace theory? In 1858, on July 1st, the secretary of the Linnean Society in London read two papers, one from Wallace and one from Darwin. The reason for that date, present at that meeting, was only Darwin. The reason for that is that Wallace was about to publish a book on the evolution of species before Darwin. And it was felt that this would be unfair and that Darwin, as the latecomer, should still be in the door. And the only way to realize that is to simultaneously read the presentation of two scientific authors, in some sense, for them. Okay? So that's the way science worked then. And as you all know, today these kind of things that somebody steals doesn't happen anymore. And the other aspect of it is, that I find remarkable, the one who came first is the one very few of us remember. It's the Darwin theory of evolution. Back again, the similarity of the genetic code is the one that is sort of the ingredient, or is the mechanism by which species evolve, life evolves. And two proteins of similar sequence, alignment of number of residues aligned versus sequence identity, have similar structure, have similar function. That means that a lot of the variation that we can see, single residue variation, doesn't matter. For instance, if I have 67 residues, or if I have 100 residues aligned, more than 33 are identical, then essentially the two have the same three-dimensional structure, meaning you can change 67 of the 100 residues, right? So most changes do not matter. Is that right, that statement? Who says it's right? So again, the statement here, 67 out of 100 can be changed. You're still in the blue region. That suggests that most, 67 is more than 50%. Let's just look at the statement for all the proteins that are 100 residues long, and all the proteins that are 100 residues long. Again, this is a small subset, we know all of that, but given that simplification, my statement is most variants do not have an effect on structure, right or wrong. Who says right? I know everybody is going to say, I don't know. You have to choose one of the two. So everybody has to say one or the other. Wrong. Yes, 3, and everybody else says right. Come on. If you have two choices, then 1 minus it is the case. So why do you believe it's wrong, the 3, the minority of 3? Anybody? Because you had to do something and it was a random fit? Anybody wants to say something about it? So the minority is right. Again, I show you something where most you can change and you're blue. Still, I make the statement that this is not representative of most changes. Why? Can anybody imagine that? Essentially what we see here is a result of a long selection procedure. What I see are things that do not matter too much. a change leads to an effect upon function and structure, possibly a dramatic one, in fact the protein no longer functions, would be one way of a dramatic effect, then this protein and possibly that carrier, the organism, will not survive. So that change does not have an evolutionary advantage, but has a lot of evolutionary disadvantage. Now what we see here is what survives. What is selected is what doesn't have an effect. In fact, what we see is the result of selection for the unlikely event. A neutral variant is relatively unlikely, and that is exactly what we see. We see the ones that either affect function in a way that helps me, or helps the carrier, or it doesn't help at all. So it's completely neutral with respect to it. So a variant that happens at some point, sometimes they are compensated for, so there's a second variant. But we have a very skewed picture of the reality. That's important. Now this is also important because when we develop methods, what we want to develop is methods that in fact see these unusual changes that do have an effect. Okay? The most extreme ones. So again, you're aware of the way chromosomes, the 23 human chromosomes are rolled up, form some structure that leads to the rolled out DNA. The DNA itself has some sort of structure, genes on it, for instance, and to just look at the level of the chromosome, there are different types of variants, and they all were presented by you on last Thursday. So there's an insertion here of a part, there's a deletion event, and this is most easily seen on the level of the chromosome really. You have duplication, you have an inversion, that's the only part we didn't talk about on Thursday. You have some translocations, they relate to something that we had before. In some sense, this is a subset thereof. And then when we go, and then, oops, on the level of the individual gene, we get copy number variations, we get mutant copies, so we get changes of the local things, and we get to change of the way introns are connected, we get the changes in the way, in fact, the protein is expressed. Sometimes entire introns are skipped. Sometimes the start codon is affected, so the whole thing is a so-called nonsense variant, so the whole protein, in fact, in this particular case, is not expressed. And then there's an effect on protein. There's an effect on non-coding long RNAs. So what I'm going to talk about only are really things that change the amino acid sequence of the protein. Okay, SNP, again, typically defined as single variants in the nucleotide sequence that occur at a certain frequency in an organism. We want to see the effect on structure and function. We want to see how the phenotype, the genotype changes. When we look at this, where we study variants, single variants, then we can see things about population. And here are a few examples. I briefly discussed about them before. Let's get back to the story. So non-coding, coding, trivial. So this variant here is not changing the protein sequence. The next one here is chosen such that it is synonymous, so it's also not changing the E. The third one, in fact, this GAA and is in fact coding for the E, as is GAG, but AAG is not. It's coding for a K. So this single variant would in fact change the protein. Now, let me just bring up another issue here. When we look at one particular position, there are 19 non-native residues, or 19 non-native amino acids. I have 20 amino acids. I can replace the native one, the one that I have in the protein, by anything else. They are not all SNP possible. Do you understand that lingo? SNP possible variants versus SNP non-possible variants. That was part of the exercise, wasn't it? Didn't you present SNP possible? You can't remember, or you didn't? We didn't. You didn't. Can you make any meaning that I put the word SNP possible in front of you? Any idea what that could possibly entice, mean? And I've put it versus 19 non-native.
cannot get any of the 19 out of 1 nucleotide.
You cannot get all, exactly. That is exactly true. This is what you mean, right?
Yeah.
So some triplets you cannot reach. So in this particular triplet, I have 3 nucleotides that I can change. But with these 3 changes, I may not get all the possible residues at this particular position. And that is the difference between SNP possible and 90 non-native. 90 non-native is intrinsically something that is coined by protein scientists. So if you sit on a particular residue and you in silico or in vitro change this particular residue against all other 90 non-native, you can do that and test the effect of that. But in a genome, you cannot possibly reach them. And there's a distinction that in fact does matter, and it's something that people begin to realize only over the last 2 or 3 years. Again, typically when I talk about variants, I don't consider the frequency of the population any amino acid change. The method that I talk about, most of the work was started from Jana Bromberg. There is a classification of the relevance, the SNP risk as classification as risk in terms of disease risk. And somehow the non-synonymous, so the amino acid changing one, have a very high risk. What is above it is splice regulation, so essentially you don't create the entire protein, and nonsense, you switch off the protein. So here you affect the protein, here you affect the protein somehow more dramatically, and here you affect the protein by completely knocking it out. And it's clear that the effect is stronger in that direction. And the probability here of downstream regulation, if you hit a regulator, of course you have a strong effect, but most things downstream simply don't have an effect. This is the way to read it. When you look at the observation of synonymous versus non-synonymous in the coding regions, what do you expect to see? When I look at all the human variation, and I asked how many of those are non-synonymous and how many are synonymous? Is there any way in which you can answer that question? Do you know any simple numbers that would help you to get an estimate that in fact is different from what we observe? Well, the simplest way to look at this is 4 by the 3 possibility, 64. Of the 64, you have 20 and 64, right? So every codon maps, so the number of codons that map to amino acid is more than twice. So roughly it would be a factor of 3. Okay, some codons are used for stop and start, but roughly it's a factor of 3. Roughly, if there were some equal distribution, you would have 2 in 3 of the variants that you observe. If variants were completely random, you would expect that 2 out of 3 are non-coding. Are non-synonymous. Synonymous, sorry. I'm not changing the amino acid, right? And that only 1 out of 3, 20 of 60, in fact do change, right? In principle. It's not quite true, because the distribution of the codons, so some codons are used more often, and the number is quite different. But when we look at the observation, the different chromosomes, synonymous is in red, and non-synonymous is in green. This clearly is not 2 parts red and 1 part green. If anything, I mean, it would be tending to be the opposite. This, in fact, is not quite trivial why that is the case. In fact, it's not quite clear. Okay? There's an observation. It's the way it is. It's not, certainly not random. When we look at the ones that affect diseases, then the story slightly gets different. So most of those, much more of those are changing the amino acid sequence. As we discussed already on Thursday, the simplest idea of using a method is SIFT. That comes ultimately from Stephen Hanikoff, the Blossom idea. The work itself was done by Pauline Nick, and the simple idea is you take a protein, you find all the residues that are similar to it, then you have an alignment. For that alignment at one particular position, you ask what's the probability of changing that E to, say, a K, and you simply compile the observed counts, and you see how much the observed counts differ using some Blossom matrix from what you would expect for that observation. So do you see it essentially in the alignment? If not, then you predict it has an effect. There's one free variable in the game, which is, so given that I give you the alignment, meaning I tell you which is the threshold, the E threshold, the E value threshold, what I cluster, what are the databases I run against. I give you the alignments, I give you the counts. What is the free variable to set up SIFT? Between this idea and actually having a method, you need somehow to decide on one free parameter. What is that? Well, essentially this alignment defines a probability, okay? It's a probability of observing the mutation. So we have an E, the observation is E to K, and you ask what's the probability of observing a K in the alignment. But if the probability is 0.8, or it's 0.3, the threshold when you say this has an effect and this doesn't, that's the single value you have to optimize, okay? So you need some data set to optimize this single variable and to just make sure that in principle the idea works. Okay? It's a very simple method in that sense. Now, what is missing in the simplest idea? The point that I'm trying to get at is if we had the sequence VHLTEP, and now I have in my alignment the protein that I have are just these three proteins. Okay? In my three proteins, I see that E, and that's the point where I observe a mutation, E is entirely conserved. I have 100% E's there. Hence, it's very important to maintain that E. Do you see the point I'm after? Everybody sees it, but can somebody say that they see it? I mean, say, what's the problem here?
Every other amino acid is conserved as well.
Yes, exactly. So you need to somehow take into account that this here is much less important than if I had another one that is L, Y, I, T, E, S or something like that. If that sequence would be in there, then this sequence overall is very different from all of the others. So now the conservation becomes more important. How can I measure the difference between these two situations, whether I have that or not? Give me a hack solution. And give me a sort of clean solution, an idea of a clean solution. So obviously, proteins that differ in sequence contribute much more information. When I just look at the probability threshold at SIFT, that reality is not taken account of. How can I take account of that reality?
Only use sequences with a sequence identity nose.
That's what I would call that a hack. So you essentially throw out everything from your alignment that is too similar, and then it is already clear that these extreme examples cannot happen. A cleaner solution? Can somebody think about a cleaner solution? A weight? The word weight, how could you apply a weight?
Maybe you could use Shannon's entropy.
That's a great idea for real information content. And this is one way of putting a weight. Yes, exactly. So essentially you would weight at that particular position the Shannon entropy of that. In the simplest way, you would, for instance, say you measure the sequence identity between the two, and the weight is proportional to the difference. So 1 minus, right? So in this particular case, the contribution of this sequence would be 0. Okay? And the contribution of this one here would be 5 and 6, so it would be very, very high. Okay, the realization of that idea is essentially the original polyphen developed by essentially Shamil, or he's sort of the idea behind it, and the latest generation here, 2010, then uses machine learning. That's an important addition. It's relatively simple, and still essentially the first version of polyphen still uses only anything that is sort of related to the conservation of one particular site, with the additional benefit or the additional issue that you sort of look at the overall conservation. Now, the next level in some sense is instead to look at some other feature, and that other feature could be structure. So you could argue whether or not you're on the surface or not, and that's the tool that was contributed originally by the Molt group, and again there's a more recent version of this available now. That gives you in some sense 3 simple tools. Here's in some sense a completely different way of looking at it. The different way of looking at it comes from the thermodynamical cycle, where you have a sort of delta G or an energy to go from the unfolded to the folded state. You have an energy transition to go from the wild type unfolded and the mutated unfolded. You have a folded and you have a folded to unfolded for the variant. And essentially the idea now is see how the variant changes this thermodynamic cycle. It comes from the group of Thomas Blundell. Here is experimental data versus the calculated delta G for one particular protein. And you see there is a tremendously good fit. Now this originally was done only using statistical potentials. Marianne Ruman expanded on that, a similar idea, but machine learning. And here is another way of doing that. iMutant2, mostly pushed by Emilio Capriotti. To do something like that, again, experimental delta G, predicted delta G, but machine learned in a sense that is similar to what Marianne introduced. Josef Weizmann has a somehow a similar idea, that again, you know, in the axis, experimental versus predicted delta G, but his method constrains it by looking at the binding interfaces, by constraining the binding interfaces according to 3D interfaces, the way proteins interact. Here is an extension of the Provian, is an extension of the original SIFT. Now, one thing that is still missing in both the hack and the clean solution, your entropy idea, the idea of a weight here, is that the weight still does not see substructure. In some sense, your hack is going into the Provian direction, where you create groups, subfamilies in this tree, and one way of your hack, the simplest way of your hack, is you have one representative for each here, and then you count the probability. Now they do that slightly differently, so they argue that there is an important conservation within that subfamily. So whether or not this E changes, and whether or not that, for instance, would be conserved in a subfamily, so CYITQS or something like that, and then we have another Q, so where we begin to see that some subfamilies have the Q, some subfamilies have the E, and that effect, in fact, still, whether I go for the entropy kind of measuring, or I go for the simplest clustering that you think about, is not captured. And that's the idea, to capture, in Provian the idea really is to capture it by separately compiling the conservation for subfamilies, and then in fact you see that locally it's conserved. Okay? The mutation taster is somehow a related idea. It's a machine learning device, but that machine learning device is essentially set up with a very human perspective, arguing that what is conserved between us and other primates has a different probability of having an effect than what is not conserved between us and other primates. So now essentially what they do is they build the machine learning by having the alignment confined to very, very close primate organisms. Okay, that's the idea here. So we change the alignment. Here the idea, that goes back to Lick charge, that goes back to a method called evolutionary tree. And in the evolutionary tree, the idea ultimately is the following, that when you look at trees, evolutionary trees here, then what you see is that the residues that define the transition here, that define whether or not you go to that or that part of the tree, those are important residues. And if you essentially cut the tree at some particular point, and at that particular point you looked at what are the residues that are conserved until then and that change then, those in fact would be the kind of things that form subgroups here. And if you have these, then most likely they are functional. That's the idea behind the evolutionary tree. Now here, in the method that typically they refer to as just action, they try to look at that particular feature and use that to compile the probability of an impact on the phenotype. Essentially, how does variation change the evolutionary landscape? That's the idea behind actually writing it in a formula. Ultimately, there is a fit of a bunch of functions given this evolutionary tree alignment for your particular protein. Okay? Here is a similar idea in a more generic form. In a more generic form is again it's the same thing, and that in fact is the first. So that goes back to Chrysander, and that's the first way to sort of formulate that idea. And Alfonso Valentia, where looking at correlated changes in families. So where the NE goes to N in a correlated way, and where something is conserved, define subfamilies, define from the alignment directly things that change for subfamilies. It is this idea here in a more mathematically clean solution. You do an eigenvector decomposition. That work was really refined by Boris Reva, and now is used to assess the effect of variance, in particular also on cancer data in this inlet. What I want to point out on this particular slide here, when you publish papers, one thing you should always think about is if your paper really is successful, everybody quotes it, then give one figure to the people to take out from the paper. This is figure 1 in the paper. It could be used as a poster, and it can be used wonderfully for somebody like me to have a quick presentation of what the idea of the method is. That's the idea, some of the results. I could spend a little bit more time if I had more. Try to think about how you can do that in your paper, in your presentation. We go to the next level that again is somehow a similar idea, is the evolutionary coupling. So when we look at residues that are close in space, since the protein or this loop or whatever evolves in 3 dimensions, this constraint from the contact is going to be visible in the alignment. So you somehow will see a correlation between this column and that column in the alignment. In this direction, in some sense, this is what it evolves as a 3-dimensional object, and that you see then in the alignment. But if you have only the alignment, can you get back the structure, the contacts? That's the method called evolutionary coupling, with which they predicted membrane structure for the first time. And essentially, it is a clever way to look at the correlation of columns. Now, since you can look at this correlation of columns, you can in fact look at this correlation and ask, if you have a particular variant, what's the impact of that variant on the function of the overall energy? Okay? And that is what is underneath here. Complicated slide, I understand. Where you go with the sequence. This is all the 20 amino acids. Strong change, I believe, is in white. So where you see the least is the most dramatic effect. The least effect is blue. ultimately, what he shows here for a bunch of proteins for which we have a lot of experimental data, that his method correlates very, very well with the experimental observation. And I cannot currently remember. Wait a minute, I could easily remember. So the blue here is the method that he has. The orange is the same concept, but essentially only looking at single columns, so not the correlation between columns. So the orange ochre color here essentially is in his formalism what you would get by a clever polyphen. Okay, this is not polyphen, yeah.
Are you sure that white is deleterious? Because
yes, it seems to give the highest value in kcal. Yes.
But the white says neutral, and the black one.
Where do you see that?
Deleterious.
No, then I'm no longer sure. Where do you see that?
On the right side.
Above this.
3D structure.
Yeah, but here's
Neutral.
Oh, that says neutral?
The red dots, these are
I thought it says kcal.
Sorry?
Oh, it says neutral? You can read neutral there?
Yeah.
And I read kcal, you see? So I'm absolutely not sure. No, in fact, yeah, you're totally right. In fact, the dot, the white with the dot is there's the residue itself. So it's total nonsense. Thank you.
And the x-axis is the conserved part. If it's
no, that is the residue running. The conservation is shown here.
Yes, that's what I mean. And the red spots are equal to the high deleterious change.
Yes, but that again. So what you see here is conservation on this. Essentially, that's the sequence conservation. So if you want, this is essentially the simple polyphen or the simple SIFT. So, but this is much more similar to polyphen, and actually much more than polyphen. But it still is looking at the conservation only, while the blue is the effect over all the 19 specific variants. Okay, so the orange is not the average. The orange is just looking at a single PSSM, while the blue is looking at pairs of PSSM. So it's always asking, if I have residue I and I have residue J, and I look at the two PSSMs, what's the probability of I and J? So of all the 20 possibilities, so it's a 400 matrix that is actually looked at. And that is the blue, and they are highly correlated. And this is in fact the blue here and the orange in some sense are whenever, so the values here go from 0 to 0.9. Anyway, you see that better than I. And you see whenever, there are exceptions, but mostly when more than average is, for instance, this one here, the blue, and for this one blue, you see that the orange is also relatively more than average. So in some sense the orange and the blue, in this sense the orange and the blue correlate, and here immediately you have an exception of that, and there's a reason for that. But the method can predict the effect. It can predict the effect that is correlated with the experimental effect. And in fact, this experimental effect is the 19 non-native here. It's a proteomic view. And it can, more than anything that I've seen so far, specifically look at pairs. So this could capture a variant, or at one position you have a V that causes the V to K that makes a problem, and can be compensated for at some other position. And that could capture that. So for pairs of mutants. Okay, let's get back to the data story. So when we started with this machine learning, trying to put machine learning into the equation, then somehow the hope had been initially that we would be able to integrate different effect on different levels. So on the level of the molecular detail, where you have delta delta G of binding, so you have a binding partner, you change the binding energy to that binding partner, and on the level of the organism. So there's some effect on the entire organism. And we had hoped that we can sort of integrate all of these data and get a machine learning device to get an average overall. It was a little bit challenging to integrate the data. There is one database that led the way. PMD is no longer developed, and some of the data that you used is from PMD. PMD has the problem of being possibly biased, so simply observation is that most of the variants that we looked at or that we accepted in PMD were of the effect type. Only 1/4 roughly were of the neutral type, and you understand why that is the case, right? Is that, do you believe that the green to red, so 1/4 is neutral, is that reality? Or is there some bias in the database that we have? So I would immediately see two reasons for bias. Can you think of any reason for bias?
It's called protein mutant database.
Yeah. Yeah, so we have mutants that are neutral, and we have mutants that are... So what do you mean by that? I believe that would be the first one, but just spell it out a little bit more. What do you mean by your observation?
Most of the mutants have to be effective.
Why? Have to be. It says who?
Because there wouldn't be mutants if they would not have an effect.
So strictly your logic is not applying, right? So you just discussed all these 14,000 away. Okay, 40,000 is only 25% of my database, but you just said 100% have to be mutant. I have to have an effect. And you're on the right track, but it's more, let's call it, it's where I look. So many of those, in fact, are studied because I see an effect in some disease, or I see some effect. That's why I go into measuring the effect. That's why I go into the detail of measuring it, because I suspect that there is something wrong. I've observed a disease that implies that site, or that protein, blah, 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 right? So there is a selection bias. Simply many sites that are chosen because there's an effect. The second one has to do with a slightly different phenomenon. And that different phenomenon ultimately is about negative data. You don't publish negative data. So it's much simpler to publish that something has an effect than to publish that it doesn't have an effect. You could have overlooked. When you say it doesn't have an effect, then essentially what you say is it doesn't have an effect on your essay. Doesn't mean it doesn't have an effect at all. But if it has an effect on your essay, then it has an effect on your essay, right? That's a clear-cut statement. When you say no effect, you always have to say on my essay, and this is easy, not as easy to sell. That is another reason here. Ultimately, the question now becomes, if you do machine learning and you have this imbalance, is that going to be a problem?
That depends on your machine learning device.
That is certainly one correct answer.
So an SVM would have no problem with that, I think.
Okay, let's begin with an SVM. And you gave the right answer. Just as you by now know, I haven't learned when this lecture starts yet, but you have learned to read me much better than I have learned to. Anyway, so let's work out why your answer is wrong. is what I'm trying to get at. The SVM would, your answer is right. It's just how I can find something wrong in your answer. Let's put it like that. So, what do you believe would happen to your prediction if you start with this as a training set in an SVM? So what I'm sorry, I'm unclear about my question. What I'm trying to get at is the SVM will learn something about that distribution, and the probability that for your next data set you will get something like 1/4 is neutral, that in your next prediction you will have a similar circle, a similar pi, is very high. So then SVM will not only learn the distinction red and green, it will implicitly also somehow learn the percentages here. It is more likely to classify something as red as effect than as green as neutral. Simply because it's sort of trained on that. Now, that implies this is okay if that were the distribution you expect. If not, then that may cause damage even to the neural network, to an SVM. You're completely right, some machine learning devices really have much more difficult issues with an imbalanced distribution. And an SVM, your answer was right, an SVM in principle could handle that, but it's not clear that you want it to handle that, because there is a bias that is going to be imprinted. One way, in fact, to get that away, if you assumed it would be half-half, is to over-represent the green, the negatives, the neutral ones. You simply do this 3 times. So you have 3 copies of the same data. It's not quite doing the trick, but it tends to put the SVM or neural network, that is more important for neural network, into the direction of, if your assumption were that it's half-half, then the likelihood that you will get something that classifies half-half is going to increase. Okay, so that means that whether or not you wonder whether the distribution is right has to do with your expectation. And that means you have to find a way to somehow get an idea about what you expect. Is that what you're going to expect? And for this particular case, we don't quite know. All the experimental data tends to say it's something that is, the red is a little bit more. It's more half-half. But red is a little bit larger, so more effect than neutral overall. That's the sort of result. It's one result from experimental measurements. So how could we now, in silico, there are experimental reasons why you don't see green, why don't you see neutral variance? How could you do it in silico? We discussed it on Thursday. You essentially can look at enzymes that have experimentally proven similar functions, provided those are similar enough in sequence that you can rule out that you misalign them. Anything where they vary, they do the same thing, your alignment is right, so anything where they vary does not affect their enzymatic activity, because you measure that they have the same, right? In this particular case here, that contributed about 26,000 and sort of evened out our set. To in fact, in this particular case, have a slightly higher set of neutrals. Looking at the performance, then we saw that in fact we looked at the differentially in different classes of buried, intermediate, exposed, effect, neutral, and you see that these pies differ. The pies differ in terms of neutral effect here, and I talked about that. This again is something that you can use as an input signal or simply as a bias that you will be aware of. Diana Bromberg did these developments, the way she did the machine learning, simple in this particular case neural network. The neural network in this particular case has an additional set of units that reflects that you have a change. So you want to predict whether or not the proline at the central position is involved in a change, but then you have to also put the y that the proline changes into. And then, in fact, what we need to replace now the system by, oh, I'm sorry, a mistake here. Why is that not? Ah, here at least it's cleaned out, but it's still not the point. So in the regular networks that we spoke about so far, the neighbors, this is for the protein interacting, not interaction, the hotspot prediction. All right? And in some sense, we always knew what the next one would be in terms of its interaction feature. That's different here. Because the next one, we have, in fact, anything that relates to the output only for one residue in the center. Okay? That's a limitation of this issue. So the degree to which we can use consecutive information, for which we can use the fact that there's something next to it, is really only sequence. There's no more correlated effects that we can get into the system easily. An advantage, or another thing that we have to do, or let's get back to the feature issue. So there's a bunch of features. We want to look at biophysical features. We want to look at sequence. We want to look at alignment. We want to look at binding sites, and we want to look at some aspects of structure here. And in fact, residue flexibility is, I didn't say that. On top, if available, we want to look at some annotations that talk about binding sites, that talk about hotspots. Again, I said that before, we have this neural network as an input. There's a unit that is coding for effect, there's a unit that is coding for not effect. It goes from -100 to 100, this score. This is the 100 unit, this is the -100 unit. What about the overall performance? Overall, SNAP on 4 particular protein performs relatively well. That's Q2, overall accuracy, how many of the residues are correctly predicted, compared to polyphen and SIFT here. Overall relatively good. SNAP S3D, the one that uses structure, is only for 1, because only for that the structure was available. There is differential behavior for residues that are buried, exposed. Here, this is the coverage accuracy for the neutral. This is coverage accuracy for the effect variance. And you see that for the neutral, exposed is the lowest. For the effect, exposed is the best. Then there is the difference between the two units that gives you the reliability index. So both of these predictions essentially would be effect, because this is the highest unit. While this here is a stronger prediction than that, the second prediction. We can use that to define a reliability index. Higher reliability index means higher reliability means more likely to be right, as you see, for effect, not effect, for neutral effect. To begin with, for all residues you predict, and this is now effect variance, you predict the performance of 77%. For all of the ones that have an effect, if you predict for every single residue, 77% of the effect ones are right in the data set that we had. If you allow yourself to look only at some of the strongest ones, sort of 50% strongest ones, the top 50%, then for that subset, the prediction accuracy becomes 87 or 88. And that is something that you get out of the prediction method. So to do this, for your prediction, you don't need the observation. You can simply look at the strength, right? And that allows you to simply look at the best. Now, as a developer, you always have to somehow come up with this curve. You have to always prove that better predictions or stronger predictions, in fact, have a higher level of accuracy. Most of the machine learning devices will do that, right? Just put the curve, because users in the future, they may only use your device for one particular variant. For that particular variant, they need to be able to say, okay, my situation is here, so that's what I expect. If you are the user and you have a very strong prediction, then you're not down to the default level. Only when you have a very weak prediction are you to the default level, and that's important for you to know. In fact, these are cumulative. You also have to put the non-cumulative. Now, another question is, so far what we look at here is, are predictions that are more reliable, that are stronger, better? What about effects? So what about stronger effects? Are they predicted better? Probability is high, because stronger effects typically are more consistent. And typically what is more consistent is what the machine learning learns. Right? Here, stronger effects in red, strong neutral green, and somehow effects in between blue. And you do see, so on the right is effect, on the left is strongly predicted neutral. So clearly, the first version of SNAP separated out some level of strength of the effect. Let's zip through. You can use that to annotate proteins. And let's go to this level here. Overall, I briefly introduced that already. Overall, again, in terms of Q2, how many residues are correctly predicted either of the two states, effect or not effect? SNAP 1 from Jana, polyphen, SIFT. These, again, this is one single parameter. This is one parameter with some clever math or averaging done over it. And this is a complicated machine learning device. Now, in terms of, yes, the orange one here is higher, but you may argue the gain is not that dramatic, not that impressive. And that is true. When we look at more detail, we see that in particular aspects of the equation or particular aspects of the story, SNAP in fact improves significantly. and it for instance, is much better in the neutrals. So there are aspects in which I can see that it improves much more than this little difference. But let's look at something else. The something else is, let's just look at cases where SIFT and polyfen, and this is polyfen 1, SNAP 1, where SIFT and polyfen agree. Okay? Let's just call everything where any two methods here agree simple. Or let's call anything where polyfen and SIFT agree simple, because we don't need a new method for that. And let's look at all the ones where they don't agree. Okay? For all the ones that they don't agree, now SIFT and polyfen, again polyfen 1 has changed with polyfen 2, becomes less accurate than random for the data set that Jana used here, which is an unfair comparison, because simply you throw out everything where they work and then the performance is very bad. So that explains this reality. But SNAP on that data set, it performs, remember the original value was here, so it clearly is less than the original value, but it does in fact gain much more for the ones that are non-trivial. Max Hecht got into SNAP 2. SNAP 2, the idea essentially was we have more data, for instance from disease, and that is the major addition of data here from OMIM type of diseases. He did not increase the neutrals, which led to a situation in which now the disease data, the effect data is more, and that is in fact why SNAP 2 has a shift toward predicting more of an effect than SNAP 1. Not because we believe this is more the reality, but simply because somehow we did not do the other end of the equation. This is an oversight, we just didn't have enough time. Also, Max felt that we should use somehow contact propensities and more detailed features of structure, contact propensities, correlated mutations, low complexity regions, disorder regions, and binding site predictions. All of that should go in, and we should have a method that in fact works without using alignment at all. We know that alignment is the strongest signal, but we want to have a method that doesn't use alignment at all, just to see what of that prediction is explained by the alignment. So if I differentially use the one with or without alignment, everything else is the same in the method, I know I can gauge the effect of the alignment on my prediction. And then I want one that has nothing to do with disease data. Simply, and I want to have another version that in fact uses annotations if available. For neutrals, and effect variants, you see the dark blue is SNAP 2, is always better than the light blue. In this particular case we only show SIFT, and the version without alignment does much better than random, but it is really poor. It's much worse than SIFT, for instance, in this scenario here. The one where we don't have the disease variants we didn't put in here. Now, here's another way of looking at it. Neutral variant. Many users of prediction methods say, well, I have SIFT, I have polyfen, I don't know which to use. The way I'm going to use both of them is by saying whenever they agree, then that's what I accept. That is this point. We simply implement the same rationale into an algorithm, into our data set, and whenever they agree. Now, you see that at that particular coverage, so when you, for an equal number of prediction, the blue here, the original SNAP, is actually better than that. Not much better. The error bars are shown by the thickness of the line. So the line is above the point. Here is the same story that I showed before, but it's done for human, and now this is done for polyfen 2. But it's still true that SNAP 2 is better than polyfen 2, is better than SNAP, and they're all better than SIFT for the variants that are predicted by other methods equally. So for variants that agree between two different methods, thank you. Now, for this particular data set here, SIFT no longer is below random. This used to be below random, but polyfen 2 is really much better. But SNAP performs still very well, and there's this relation to reliability. So now here again, I said that before, the version of SNAP 2 now can predict for proteins for which we don't have any alignments. And for some of the cases, this is very important. So the next issue really is going beyond singles. What could I mean by that?
To include other sequence variants than single nucleotide polymorphisms.
Such as?
Insertions, deletions.
That's a great idea. And that's what Provian does. And I believe this is something that we should work on. That we should add something that is, I believe, you could argue is more machine learning based than Provian is. We don't do that at this point. There's another way of interpreting this segment. That's true. This one of the stories, let's call it a master thesis, that is open.
You could check for combinations of SNPs to look at complex traits.
Yes. Well, that's a good definition. So anything that is more than one is already more complex. And that's why I call them complex traits. Yes, and this is in fact exactly what the method from Thomas Hopf does. And there's a master thesis that somehow gets in that direction at the moment. There is something, maybe there's some more room. And I'm also not going to talk about that today. What else could it be? So the simplest way of going to more than just single ones is you just repeat the same thing. So you have a particular residue, you change it, you can snap predicts the effect. What if I did that for many times over? So not only one, so at every single position I do 90 non-native or SNP possible. Okay? And then I simply average and see, do we have places where they all agree? And this is one scale here where you see there's a region where essentially every prediction says effect. Then there's a region where everything is sort of not really effect. And interestingly, this is exactly the P53 functional region. Nothing else put in here. We see that it correlates with binding sites, and in this particular case, we sort of see the more red, the more you're close to the binding site. It's just 3 different ways of averaging over what I call an effect. And there's a story that is open here to do the effect variant a little bit better. You can then apply that thing to, for instance, in this particular case, the melanocortin receptor, transmembrane protein, and annotate the predictions and the observations of what causes an effect that is related to obesity or to diabetes in this particular case, for both of those. And then what we did is, where are cases where we predict a strong effect and there's no experimental measurement? So essentially as a direction for experiments, so this was done with collaborators, published here in FASEP with Rudy Labelle, and they in fact measured some of these predictions, and several of these led to papers. Now the unique reality that you can create, looking at 2 similar receptors, the overall percentage sequence identity is a ballpark of 50, 2 melanocortin receptors, both human. Now let's just look at sites where, and I lost the annotation already in the one before. What I want to look at is green and red. Green and red. I'm sorry, I should have marked it. So here we have a site. This is a leucine. So we have 2 receptors that overall have a similar function. In detail, they have differences. In this overall, more than 50% sequence identity, in this overall alignment, we pick a site where they have the same letter. Now, I do a 90 non-native scan for both. And I see that one of these 90 non-native scan here predicts no effect, and one of these predicts strong effect to any kind of mutation of that site. Meaning, I would argue these 2 differ. Meaning, I have a possibility now to go at a site where I have the same residue and differentially predict something. So I can say this is a site that has to do something with the functional difference between the 2 proteins, although I in fact have the same residue. And that's a remarkable reality. There's no tool in bioinformatics that can do something like that. Any tool will always look at identical residue, identical thing. There is no way in which you can sort of differentiate between, in two proteins at least, where you overall believe they do the same thing, differentiate at a position where you have the same residue, the same amino acid, similar alignment, still the method predicts the differential behavior. And that's remarkable. And for some of these cases, we traced those and they made sense. So essentially what the message here is, we can look at, that shows the sequence running here. That's the 20 non-native predictions, superpositions, variants, where more red in this particular case is effect. You have places where you have a lot of red, you have places where you have a lot of green. Now you look at the ones that have a lot of red, you sort of filter it by blossom, then you filter it by the ones that are predicted to be in contact, and then essentially in this particular case you get exactly to the binding site. No further assumption. You don't know the structure, you don't know anything about where the binding site looks, you just begin with the red. You filter according, so the way this blossom filter here is essentially what you're doing is, if blossom says it should be neutral, yet you predict an effect of that variant, then that must be something important for function. That is this step, and then you simply look at a contact predicted and you get to the end. Okay. I believe I knew this wouldn't fit, but it fits much less than not. Let me get just to one point in the end in the analysis, because we repeat something that we already have briefly discussed, which in fact is the story where again I have the SNP score for severity. It's a cumulative curve. I asked what fraction of my set is predicted at a score higher than something. Again, high severity, high reliability of neutral. The set that I'm looking at in red is the training set. So at the threshold of 50, about 20% of the residues in my training set that are known to have an effect are predicted to have an effect, and so forth. Black is OMIM. Those are the variants that where a single variant causes a disease. That is above the training set. Okay, I briefly mentioned that. Now, is that enough now to look at a variant and predict that it is an OMIM type of variant? So could I then essentially take this out here, the black curve, and say whatever is in the highest point here is going to be an OMIM-like variant? Can I? So I cannot. Why not? Because ultimately, every pair in us has 20,000 variants. Yes, this black is much above the training curve, but in 20,000 there are always some there. And we are not all running around with killer diseases. Okay? So, although the OMIM variants are clearly predicted right overall, for the individual variant, I do not know whether it's one of those. Because too many of the variants that we observe between people, in fact, are predicted with very strong effects. They may have effect, so now the story gets more complicated, and maybe that's the level where we should get at the network level. But again, so what this one here shows is the same thing as before. In this particular case now the red is OMIM here, that's the training set, and now we have a version of SNP2 that in fact has not only used the disease data. How about diseases in animals? And the answer is that the effects of disease in animals are even higher, typically because animal diseases are more severe in order to be considered diseases. Sorry for this extremely condensed end of it. Thomas is one of the people who is going to bring it to the level of pairs. A lot of what I presented today was from Max, Tobias, Tanya, Arthur I zipped over it. Laszlo and Guy I zipped over their part, and the disorder part here of Esmeralda. There are many people to thank. Thank you for your attention for the lecture. Sorry for the fast end of it. Thanks.